Thanks, Malcolm, and good afternoon to everyone. Okay, so today I've divided my talk in two. I'm going to start by talking about the effects of climate change, specifically temperature, increases in temperature and acidity on early life histories of corals. And then I'm going to present some work I did with Joana Figueiredo, looking at the effects of temperature on local retention of coral larvae. So climate change is going to affect organisms in two major ways. There's going to be increases in temperature, and in the oceans there's going to be increases in acidity. The effects of temperature are pretty well documented. It's known that increases in temperature increase metabolism, and that speeds up most developmental processes. Increased temperatures also produce toxic oxygen radicals in the cells, and so you have increases in cell death. So again, a fairly well-established pattern of higher rates of mortality at higher temperature. Acidity, in fact, has a different effect on rates of development. You can demonstrate this for yourself by, if ever you've got a coral larvae, just pop it on a microscope slide and add a little bit of carbonated water. And it's quite extraordinary the effect that this has in slowing down the speed of a coral larvae. The effects of acidification on mortality, again, I think are less well documented. Adjusting to pH is going to be an energetic process, and therefore in critical life stages such as coral larvae, that may come at a cost of increased mortality. This is a diagram illustrating the life history of a coral. We have a sessile adult in the left corner there. They release their gametes into the water column. They're fertilised. They develop through embryogenesis into a motile larvae, which swims down to the bottom and settles, producing a juvenile and completing the life cycle. This is an illustration of the process of gametogenesis. You've got a coral egg, and in the second panel there is a cleaved embryo that develops through a number of stages to a motile larvae there at the bottom right, and that can take between 12 and 36 hours, just depending on the species. The critical stages in this process that I've identified and that we looked at in our experiments are fertilisation, the rate of embryogenesis, so the time it took to complete embryogenesis, metamorphosis, so we estimated how effectively coral larvae could metamorphose at the end of this process, and we also looked at coral larval survivorship. We worked with a couple of species. This is Acropora tenuis, and this is Acropora millipora, and we did 4 different experimental treatments. We had an ambient treatment where we work at Magnetic Island. The temperature at the time of spawning is typically about 27 degrees, and we used an ambient concentration of carbon dioxide of 380 parts per million. We also had a high temperature treatment, which is a +2 over our ambient, which is approximately where we'll be in about 50 years' time. We had a high CO2 treatment, 750 parts per million, again, approximately where we'll be in about 50 years' time, depending on the scenarios. And then we had a treatment that combined high temperature and high carbon dioxide. We used a setup first put together by Phil Munday. Essentially, we just mix pure CO2 with the atmosphere, with air, bubble it through water to produce our acidified water, and for that temperature treatments we just use heaters. So again, the first variable or critical life stage we looked at was fertilisation. We put a number of eggs and sperm into replicate treatments, place them in each of those treatments, and then score after 2 hours the proportion of those eggs that have successfully fertilised or cleaved. And as you can see from these data, we did this 4 times. We did it with Acropora tenuis, and then we did looked at 3 separate cohorts of Acropora millipora. And you can see from these figures, the grey bars are the ambient PCO2, and the black bars are the high PCO2 treatments. And so you can see that in all 4 of these assays, or in 3 of these assays, there was no effect of either acid or temperature on rates of fertilisation. only in the third assay for Acropora millipora did we see reduced rates of fertilisation at high temperature. So not much effect of any of these variables. And so the next variable we looked at was time to motility, so the time it took for the embryos to become motile. Here on the y-axis is the average time to motility, and again we did this 4 times. In the first 2 experiments, we saw an interaction between acid and temperature. So as you can see at the 27-degree treatment, the larvae are taking longer to become motile in the high acid treatment in that black bar. So that's what you would expect, the developmental process being slowed down. But at 29 degrees, we saw the opposite effect. It was actually, the time to motility was reduced in the high acid treatment. In the second panel, again, an interaction between temperature and acidity. In the 27 degrees, we've seen, again, a speed up of the rate of development. Larvae are becoming motile more quickly at high acid levels. At 29, there's no difference between those treatments. In the second assay we did for Acropora millipora, this was one of the few occasions that we actually did see an effect of ocean acidity. So if you combine those 2 grey bars and compare them to the combined 2 black bars, you can see again that larvae at the high acid treatment are taking longer, are becoming motile more quickly than at the low acid treatment. And finally, in the last assay, there was an effect of temperature. So larvae in the high temperature treatment are becoming motile more quickly. Next, we looked at the metamorphosis, so the proportion of larvae that we introduced to conditioned tiles that successfully completed metamorphosis. And again, in the first assay, Acropora tenuis, we saw an increased rate of settlement at high temperature. But in Acropora millipora, we saw a reduced rate of metamorphosis, which is actually the next bar across at 29 degrees. So again, very variable effects. Finally, we looked at larval survivorship. So those lines, the values in the lines are the median number of larvae alive through time, and the error bars are 95% confidence intervals. And again, we did this with each of these study species. For Acropora tenuis, the top panel, there was no difference in any of our treatments. In Acropora millipora, we saw an effect of temperature, so that green and the red line in the bottom panel are the high temperature treatments. So again, no effect of acidity on larval survivorship. So just to summarise, the effects of climate change on the early life histories of corals. The results are very variable, so it's very difficult to make any generalisations. But I would say that there was no strong evidence for an effect of acid, so in particular at these levels. So I think the conclusion is that at least the direct effects on the early life history stages of corals, we're unlikely to see any major ecological impact, at least at these levels, so over the next 50 years or so. So next I'll talk about how climate change, more specifically increases in temperature, might affect patterns of dispersal. So again, we know that temperature increases rates of larval mortality, but in contrast, it increases rates of development. So the larvae that are produced on a given reef, a lot more of them are likely to die in a given period of time, but more of them are likely to become competent more quickly. And so the net effect of these 2 processes is not clear. And so that's why we need some experiments to tease those things apart. And so this is work I did with Joanna over in Japan, and we have our temperature baths, and we introduce coral larvae into small glass jars, which we then place in the temperature baths, and then we count the number of metamorphosed corals on those conditioned clay tiles through time. We also independently made estimates of larval mortality by, again, just placing larvae into the jars and counting them on a daily basis. And then we used this model of Sean Connolly's to fit curves to our empirical data on changing competence through time. Those curves allow us to estimate a number of parameters that we can compare among our treatments, the most important of which is TC there on the left, which is the time delay until these larvae can become competent to settle. So the top panels, top 3 panels, we did this with 3 species. The top 3 panels represent the survivorship of these larvae in the different treatments, coloured by the lines coloured by the treatment. So we had an ambient treatment of about 27 again, 29 and 31. And so you can see that the red line being the high temperature treatment, the mortality rates are higher at high temperature in 2 of these 3 species. The bottom panel is the proportion of larvae competent through time in these 3 species. And again, you can see probably most clearly in Acropora millipora, which is the bottom right panel, that the time delay to competence has come forward about 24 hours. So larvae in these treatments, in this +4 treatment, are settling a full day earlier than they are at ambient treatments. So we've got these 2 competing processes. We have increased mortality, but we also have larvae developing more quickly. So what effect will that have on patterns of retention? So what we did to look at that was we used that data on mortality, we used that data on change in competence through time, and we looked at how the relative increase in self-recruitment would change as a function of the average retention time of particles on the reef. And again, it's probably most clearly represented in Acropora millipora, the panel on the right. So you can see that on reefs with an average retention time of a day, the increase at +2 degrees C will be approximately 3-fold increase in rates of self-retention. At +4 C, we're going to get an increased rates of self-retention of a 5-fold difference. So 5 times more larvae are going to remain on those reefs. The effect diminishes whereas as average retention time increases. But average retention times on most reefs are on the order of 3 to 5 days. So in the vast majority of reefs, we're going to see quite dramatic increases in the proportion of larvae retained. So what are the potential implications of that? I mean, certainly stock recruitment relationships are likely to be much stronger, and that's generally a good thing. Rates of recruitment are likely to be higher, and therefore reefs are likely to be more resistant to disturbance, at least at the local scale. But increased rates of self-retention mean that less larvae are going to be leaving the reef, and so patterns of connectivity among reefs are going to be reduced. That's not necessarily a bad thing, but it certainly means that we need to think about how we manage the parks. For example, we might need to increase the size of protected areas or adjust the spacings so we can compensate for these reduced levels of connectivity that we're going to see as a consequence of increases in temperature. And with that, I would like to acknowledge my supporters and thank you.